Okay, guys, quick video again on our Roman numeral 1 from our outline. Here is our outline just to show you on cellular transport. It's our introduction. We want to go over the difference between homeostasis and equilibrium. This is on your classwork tab, by the way. Feedback mechanisms and cellular boundaries. Okay, so here we have the difference between homeostasis and equilibrium. Homeostasis is actually the regulation of stability. It's a big picture item. It's when you're talking about the whole organism, for instance, and how he maintains that balance of many, many things, from water to body temperature to blood sugar levels to anything you can think of. And equilibrium is more of like an isolated situation that you're looking at. And you're looking at that balance or stability. Like, is your body temperature regulated properly? Is it in equilibrium? Is your blood sugar at equilibrium, what it should be, okay? But homeostasis is more how that is regulated, okay? And it's more big picture. Then there's feedback mechanisms. So if any of this gets off balance, right, then how do you get it back on? Well, if you get off balance, your goal is to get back to normal, get back to that homeostasis. And there's two ways that you can achieve that. You can do it through positive feedback mechanisms or negative feedback mechanisms. Positive are going to enhance something to reach that goal. And good examples of that is fruit ripening. Okay, because fruits will get riper and riper and more ripe until either it attracts an animal to eat it or it falls off of the tree or the vine and releases the seeds that are within it. Okay, that's one example. The other example is childbirth. Okay, childbirth. When it comes to childbirth, a woman's contractions will get stronger, and more frequent until that baby is born, okay? And both of these are examples of positive feedback. Positive doesn't mean it's a good thing, but it does mean that it's going to enhance something, so think positives. Get more until a goal is met. Negative, on the other hand, does not mean a bad thing. It means it wants to hold something steady. And the best example of that is body temperature. Body temperature. We all know that we have like an internal normal body temperature of about 98.6 degrees Fahrenheit, that is. And our body temperature, if it gets cold outside, we shiver, which keeps our body temperature maintaining that 98.6, or we put on a jacket if we're smart. But body temperature on a hot day could go up, right? So what do we do on a hot day? We sweat, and that cools us down. So that's one example of negative, where it's trying to maintain a steady, even keel, like a certain set point. Another example of negative feedback is blood sugar level. Blood sugar. Okay. When sugar is in the blood, it needs to get into the cells where it's going to be utilized, and the energy is going to be extracted from it. And there's one substance that's produced by your pancreas, actually, that does that. When blood sugar level is high, so if this is high, then your body will produce insulin to send that into your cells. And then it goes into cells. But if your blood sugar is low, then your body will produce another substance from the liver called glucagon, that will release sugar from the liver into the blood. So glucagon will raise your blood sugar. Insulin will lower it. So when your sugar is high, insulin will lower it by shoving it into the cells. When your sugar is low, glucagon will release it from the liver to rise it back up so that it maintains a steady level. Okay, and insulin is produced by the pancreas. Glucagon is also produced by the pancreas, but it gets the sugar out of that. So when you don't eat, okay, you have low blood sugar. So what your body actually does starts producing glucagon and steals some of that sugar from your liver. Later on, you're going to have to replenish it. So when you eat, you not only have to eat to get the sugar into your blood, but you also have to replenish the sugar that was taken from your liver. And then after you eat, you produce insulin, which shoves that sugar in your blood after it's digested in your blood. Then it shoves it into the cells where it will actually do its job. Okay, and so that's the first two things on our cellular transport outline, homeostasis and feedback. The next thing I'm going to bring up is this diagram of the cell membrane. So push this aside. And this is letter C, all about cell boundaries. And when we talk about cell boundaries, it's all about the cell membrane. So you have an Edpuzzle on this, and you have this diagram here that's also on the thing. But I want to go over these 4 points, these 4 molecules in there. Now you'll notice protein, 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 and here's another protein here, and here's another protein here. We can consider them all proteins. Phospholipids right here, okay, phospholipids are little balls with 2 tails. Okay, they have the head and the tail. The head is the phosphate group and the tails are the fatty acids. The head likes water, the tails don't like water, and there's a bilayer where some point out and some point in. So say this was the inside of the cell and this is the outside of the cell. We have phospholipids pointing into the cell, phospholipids pointing out. Phospholipids are the barrier molecules. They're the barrier molecules. Okay, the barrier molecules. They're actually setting up the actual cell membrane, okay, the barrier, okay. Proteins are the true regulators. They're the true regulators. They're going to actually do, when we say that the cell membrane regulates things going in and out, it's really all about what the proteins say. They're kind of like the boss, okay? Then there's cholesterol, and you can see little pieces of cholesterol here and here. They're embedded in those phospholipids, kind of keep those phospholipids like at a safe distance, kind of like social distancing, okay? So they don't get too tight. They are somewhat tightly packed, not like, COVID people, but they're tightly packed, but not too tight. If they get too tight, the cell membrane could rupture easily or something. So the cholesterol kind of like keep the cell membrane loose. We'll say the phospholipids. Let's say fluid. Okay, and we mean like kind of loose, fluid-like. Okay, and then finally the carbohydrates. That's all these things on top here, and notice they're only on the outside of the cell membrane, and they can be attached to phospholipids, and we call it a glyco. The carbohydrates are the glyco. See, glyco. Whenever you see glyco, that's it. Here's the glyco there. Okay, they can be attached to a lipid or they could be attached to a protein. Here they are attached to a protein. Here they are attached to a lipid. Another one attached to a lipid, one attached to a lipid. But they're the carbohydrates, the glycos. And they're like your fingerprints. They're the ID tags of your cells. They tell your cells from other things. Okay, so they are like the I like to call them the fingerprints. The fingerprints of the cell. They identify the cell from things that are not its kind. Okay, so just like your fingerprints identify you from somebody else, these carbohydrates identify you from somebody else, okay, in your cells. And that's the cell membrane, okay? And that's it for this video. Take good notes.